Welcome to the deep dive. Today we're tackling a really interesting topic in bioprocessing, how changing the outer layer of cells, their membranes, can make industrial biological processes, well, way more efficient.
Yeah, it's quite something.
Think about it. Producing medicines, creating sustainable fuels, this subtle change can have a huge impact. We've looked into some pretty compelling scientific case studies covering both chemical and genetic methods.
Yeah.
So our mission today is to really get into the nuts and bolts, how and why fiddling with cell membranes leads to these big improvements, and maybe find some, surprising applications along the way.
It's fascinating, really. This outer skin, so fundamental, acts as this key bottleneck or maybe a gateway. In so many industrial setups, we're basically finding clever ways to coax cells into releasing what we want or taking in what they need.
Or even just surviving tougher conditions.
Exactly, surviving the production environment itself.
Okay, let's kick things off with making cells better at producing proteins specifically, getting those valuable recombinant proteins out more easily. Now, for listeners, recombinant proteins are ones we've engineered the cell to make, often for medicine. Getting them out easily is a big step, right?
Absolutely crucial. It simplifies the whole purification process downstream. One chemical approach, seen in Bissell Sudless, uses pretty simple stuff detergents like Triton X100 or solvents. What's interesting is how these chemicals sort of mess with the membrane's lipid structure, creating temporary holes. This allows larger molecules, like the alpha amylase enzyme in the study, to just diffuse out much more readily. It's like loosening the cell's grip.
So a straightforward chemical push. What about genetics? The E. coli example, where they make more efflux pumps, Tolsi, HI-Lib, that sounds more targeted. What exactly are efflux pumps?
Think of them as protein machines embedded in the membrane. They use energy to actively pump specific molecules out of the cell, like tiny, very selective bouncers. So by making the cell produce more of these pumps, it just becomes a much more efficient exporter for those specific antibody fragments they were studying.
Got it. So whether it's chemically loosening the membrane or genetically adding more exporters, we make purification easier. Now, what about actually boosting the production of other valuable things, like secondary metabolites? Stuff like antibiotics, right? They aren't directly for cell growth.
Precisely. And often these compounds are trapped inside, or their production is limited by feedback inside the cell. The Streptomyces erythraea case is a good example here. Using surfactants or solvents to increase erythromycin production suggests that making the membrane more permeable can actually relieve some of these internal bottlenecks.
How so?
Well, if the product can leak out more easily, it doesn't build up as much inside the cell. High internal concentrations can often trigger feedback inhibition, basically telling the cell, okay, we have enough, slow down production. So increased permeability can bypass that signal.
That's clever. And genetically, they're even changing the membrane's lipid makeup in Streptomyces. How does altering the membrane itself affect what's happening inside the cell, leading to more antibiotics?
Yeah, the membrane isn't just a static bag. Its specific lipid composition, the types of fats it's made of, affects how fluid or rigid it is. This fluidity directly influences the function of many membrane-bound enzymes.
Ah, okay.
And many enzymes critical for antibiotic synthesis pathways are located right there, in or associated with the membrane. So by fine-tuning the lipid environment, you're essentially optimizing the working conditions for that crucial machinery.
Right, makes sense. So we've covered getting products out and making more of them. What about getting the raw materials in faster? Substrate uptake, that's got to be important too.
Oh, absolutely. Cells need to eat efficiently. The chemical approach using solvents like toluene or xylene to help Pseudomonas pudida take up toluene is interesting.
Especially for bioremediation, right?
Yeah.
Breaking down pollutants.
Exactly. These solvents likely make the membrane more fluid, allowing hydrophobic molecules like toluene, the pollutant, and the food source here to dissolve into and cross the membrane more easily, helping the microbes eat the contaminants faster.
So making the front door more accessible. And genetically. Engineering E. coli with new porins. What are porins again?
Porins are basically protein channels. They form water-filled pores through the outer membrane, mainly in bacteria like E. coli. They allow passive diffusion, but only for molecules up to a certain size.
Okay.
By engineering new porins, or modifying existing ones, scientists can effectively increase the size limit or change the selectivity, allowing the cells to import larger or different types of molecules they couldn't before, expanding their metabolic toolkit, essentially.
Giving them a broader diet. Cool. Okay, lastly, these bioprocesses can be pretty harsh environments, toxins, solvents, byproducts. How does membrane modification help cells just, survive?
Tolerance is key for efficiency. We saw efflux pumps for getting products out, but they're also crucial for survival. Genetically boosting efflux pumps in Pseudomonas purida helps them resist solvents by actively pumping the toxic solvent molecules back out before they cause too much damage.
Fighting back, basically.
Right. And then there's the yeast example, Saccharomyces cerevisiae, dealing with high ethanol levels during biofuel production. That's a classic challenge.
Yeah, ethanol is toxic to the yeast producing it.
Exactly. So altering the membrane lipids here, like increasing the saturation of the fatty acid tails or adding more ergosterol, makes the membrane more rigid, less fluid.
Why does that help?
Because ethanol tends to increase membrane fluidity too much, disrupting its structure and function. A more rigid membrane can better maintain its integrity and barrier function, even when surrounded by high concentrations of ethanol, allowing the yeast to keep fermenting for longer.
So quite a range of strategies. To sum up then, it seems whether we're using chemicals for a temporary effect or genetic engineering for a more permanent change, tweaking the cell membrane gives us this really versatile set of tools. We can optimize getting things out, making more stuff, getting fuel in, and just helping cells survive the job.
It really covers multiple bottlenecks in bioprocessing, all by focusing on this one cellular structure. And the benefit for everyone, really, is that these improvements can lead to more efficient, potentially cheaper, and more sustainable production of so many important products we rely on.
Definitely. It really gets you thinking, doesn't it? What's next? Could we engineer like smart membranes? Membranes that could sense the environment in a bioreactor and change their properties on the fly, become more permeable when needed, then tighten up for protection.
That's the frontier, isn't it? Dynamic control is a fascinating possibility. Lots to still explore there.
Definitely something to chew on. Thanks for joining us for this deep dive.